This screencast is going to cover active transport, endo, and exocytosis. So text references up here describe and explain here this active transport, and then we're going to work on endocytosis. There are two subcategories of that, pinocytosis and phagocytosis, and then quickly look at exocytosis as well. So previously, we have looked at passive transport mechanisms, diffusion and facilitated diffusion, and been working on those, going from transporting molecules across membranes from high concentration to low concentration. We're now going to look at this active transport mechanism, which is, again, moving molecules across membranes. But the biggest deal here is that active transport is a process where free energy, often provided by the hydrolysis of ATP, we'll talk about that in a second, is used to move molecules across membranes to establish or maintain concentration gradients. So now, instead of going down the concentration gradient, these active transport mechanisms are making the gradient. So if you look, here again is our membrane. One side of the membrane, we have molecules that are in a relatively low concentration, and on this side they are in a higher concentration. And we have a protein that's in the membrane here, and this protein is now going to be referred to as a pump. This is how we commonly refer to them, as pumps. So in an active transport mechanism, we're moving these things across the membrane, but the active part here is that they are going from a low concentration to a high concentration. They meaning the solutes here. The other active part of this is that there is going to be some sort of energy needed to do this. Now, it's not always ATP used here, but it frequently is. We'll talk about other situations where it isn't in the coming weeks. So we have these protein channels. Go back. These are not protein channels. These are pumps. Right? A channel is not a pump, so pumps do not equal channels. Channels here are typically associated with facilitated diffusion. So here's an opportunity to look at similarities and differences between these other transport mechanisms, right? That this is similar in that I'm using a protein. It's similar in that I'm moving molecules, but it's different in that I'm moving molecules from a low concentration to a high concentration. It's also similar in that typically these solutes here tend to be large, right, charged, or polar. So these things are often one of these, but could also be nonpolar. But again, the big deal here is that you're going from a low concentration to a high concentration, making the gradient, establishing the gradient. Right? Establishing, another word that will come up is a potential difference. So this concept of creating a concentration gradient, and also a potential difference, right, across the membrane. So there it is all together. We've seen this whole image, this diagram, right? Comparing, contrasting, being able to identify, describe the similarities and the differences between all these transport mechanisms is what you need to be able to do. Examples of active transport mechanisms. The most famous example is the sodium-potassium pump. Right? So a specific example is establishing and maintaining gradients is this sodium-potassium pump. So let's look at the diagram for a second. Here we have a membrane, and in the membrane we have this protein, and this protein is called the sodium-potassium pump. This is the sodium-potassium pump. Right? So this protein is a very special protein. It is able to move sodium ions as well as move potassium ions. And this protein changes its conformation. So if we start up here at 11 o'clock, right, number 1, we're going to get 3 sodiums bound to it. Sodium binds here. We use this ATP molecule, right, attaching this phosphate. This is going to, in some way, energize this protein. It gets it to change its conformation, right? So see from here to here, there's a conformation change. This changes the shape. When that shape change, it expels these 3 sodiums outside, concentrating sodium on the outside. So the concentration of sodium ions is going to go up on this outside. This is an active transport mechanism. It keeps moving sodium out of the cell. When it changes its conformation back, notice that this phosphate group goes away right here. And so then as you go here, this is another change in shape. The potassium ions now get picked up and are moved inside of the cell. So again, we're going to now concentrate potassium on the inside. This happens to just move 2 to the inside. So again, we're going to concentrate potassium on the inside. So again, this is an active transport mechanism because you're going from low to high, making a gradient for potassium, also at the same time making a gradient for sodium. You can click on that animation and it'll run it through there for you if you want to take a look at that active. So this sodium-potassium pump is an example of an active transport mechanism. Pretty famous because lots of cells have that. Other pumps that we will see coming in the future, this is a proton pump, not a sodium-potassium pump, but it's a pump. Again, this is indicating that you're going low to high concentration, and this pump is pumping what? It's pumping protons, which are hydrogen ions. So a hydrogen ion is a proton. These things are the same. We're going to use ATP to run this proton pump. And again, we're making a higher concentration on this side of the membrane of hydrogen ions, and it's lower on this side of the membrane. We will see proton pumps in cell respiration as well as in photosynthesis. So we've got different pumps, right? One specifically for sodium and potassium, one for hydrogen ions. We're going to see this concept applied. We make these gradients, and then we're going to use the gradient here. Right? So we're going to allow this diffusion of the protons back through a co-transporter molecule. So this down here is facilitated diffusion. Right? This is an active transport. This is an example. This slide is an example of coupling reactions together. Right? And this goes back to our conversation of changes in free energy and what do I want here? Energy changes, reactions, right? Negative delta G's, positive delta G's. Okay, we talked about this earlier. And so coupling the movement of those ions down gradients. This right here would be a negative delta G. This right here is a positive delta G. Making the gradient and then using the gradient. So as these things go down the gradient, we're then these sucrose molecules would hitch a ride with these protons coming back, diffusing, right, back across the membrane. So as this goes through, so does the sucrose at the same time. This is why we're co-transporting these things back in. This is passive. This is a negative delta G. But it's not going to happen unless you make this gradient over here. Right? And that's the job of the pump, to make those gradients. Again, we're going to see these specific examples in the coming weeks. Another way to get stuff across the membrane is either exocytosis or endocytosis. So in endocytosis, right? In endocytosis, the cell takes in macromolecules and particulate matter by forming new vesicles derived from the cell or plasma membrane. This is best seen by clicking these animations and or looking at them on Blackboard. I'll post them there. But essentially what's happening is you're getting a solid particle here that's going to be enveloped by the membrane, and then that membrane sort of pinches off here and pinches off here, and now you form this vesicle. And there's a membrane that surrounds this solid stuff there. If the stuff that's getting pinched in, that's getting endocytosed, is dissolved, so you're assuming that these are dissolved solutes. These are not dissolved solutes up here, food or other particles not dissolved. You get two different types of endocytosis there. If you have this not dissolved particles, it's called a phagocytosis or eating. Right? Phago, eating. If the solutes are dissolved in the solution, then it's a pinocytosis, and this is drinking. But either way, it's still the same mechanism, right? You're getting massive. You're getting lots of quantities of this stuff, and the membrane is pinching off, and you get this vesicle. Right? So on the right here, you see electron micrographs of a bacterium. Here's the bacterium right here. And this is an amoeba. This is a eukaryotic cell, right, that is engulfing it, surrounding this bacterium, and it's going to take it and stick it into this food vacuole. What's inside that food vacuole? Enzymes, which are going to break down this bacterium, nom, 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 and give it, energy, etc., raw materials to do respiration. This photomicrograph down here, sorry, electron micrograph, not a photomicrograph. Electron micrograph is showing little vesicles forming, right? Penocytosis, right? And then this is the larger, these vesicles all sort of fuse together into one larger vesicle there. Right? The opposite of that would be an exocytosis, and we're going to see this when we talk about gene expression and making proteins that are going to be delivered outside of the cell. So here's our ER, membrane-bound organelle. These are vesicles that are little membranous sacs, right? And this is the Golgi apparatus. We'll do this in more detail later. But what I want to focus on, again, is this down here, that these vesicles are sort of moving in this direction. And now the vesicle is going to fuse with the membrane and dump the contents to the outside of the cell. So these little reddish, purplish, or whatever color that is, these things are supposed to be like little proteins that are then destined to be released from the cell. So exocytosis. Click on those animations or look at them on Blackboard, and that'll put it in motion for you. And that's it. Questions? Bring them to class.